So welcome back, everybody, and welcome back to our Test Him Tackling for Fertility podcast. I'm Toby Trice, one of the founders of Test Him, and I'm joined alongside Ian Stones and Michael Clace. How are you guys doing today?
Good.
Yeah, very good. Thank you.
It's good to be back, isn't it?
It is good to be back. It's good to be back. It's nice to tune in, chat fertility, and help educate all the guys out there and support men through this unusual world of fertility that's very difficult to come by. And we just want to do everything we can to help you guys out.
Yeah, definitely.
Let's talk testicles.
Let's talk testicles. Let's talk balls.
1, 2.
Yeah.
So, today's episode on the topic of testing, we're going to talk through semen analysis, what it means, what is the terminology, how to kind of like understand it a little bit, just help educate you. But actually, there's a lot more tests out there, and we're going to talk a bit more about those other tests to help you realise your fertility potential. I think often men just go through a semen analysis, don't really understand their results properly, and then actually don't know where to turn to next. Some just think that that's it, but actually there's so many other tests out there that can really, really help out and help you understand if there are any other underlying issues. And I'm going to give an example, I suppose, if we come straight to me on my story. I think it's probably a good prompt.
Perfect example.
Perhaps a little bit ahead of describing semen analysis, but throughout my journey, I only ever had 2 or 3 basic semen analysis prior to IVF. And then I went on to have IVF and only had another couple more semen analysis. And it didn't really give the full picture. And the following advanced tests that I had, all thanks to the amazing people that I'm currently sat with, gave me more information so that I could get diagnosed correctly, then got treatment, and then went on to have success, which was really, obviously, very, very emotional for me. I'm very grateful for that. But the entire time up to that point, I was a complete rabbit in headlights. I had no idea that these things existed. So I think before we talk about those, it's probably good to start off with what is a semen analysis, what things are tested in a semen analysis, and can we talk through what the weird and wonderful words are and what they actually mean?
Yeah. Well, Michael, I think you're the one for the words, aren't you?
Oh my goodness.
Your oligos and your... Well, that's the only one I know. That's the know.
Well, there's lots of funny names, that's for sure, for abnormal semen analysis and things like that. But I think probably the first part is understanding some of the components of it, really, because they all add up to form a picture of really what is going on and whether there is good fertility capacity or integrity, I suppose. So I think the easiest way to describe it is it's split into 2 ways of looking at it. One is like a macroscopic view, sort of a wider view. So some of the elements that you'd look at is things like the volume. Volume is quite important. Things like the colour, the acidity, or almost called pH. And then other things to look at would be viscosity. So it's how thick or gloopy the sample is, because the more gloopy it is, the more difficult it might be that the sperm can move in that sort of substance. So those are the sort of like what the eye can see just looking at the sample itself and what you can measure at the top end. And then more importantly, I suppose, for looking things from a clinic point of view, they'll be wanting to look at microscopic view of the world. So that is really looking at counting and what is moving and so forth. So things like that are things like concentration or the count, how many sperm are actually floating in that volume of liquid that is produced as a specimen. But then looking at the motility. Motility is really important, and often it's split down into 3 components. One is, are the sperm moving at all? So are they non-motile, or are they motile? Is the sperm that are moving? They're probably a little bit better than the non-motile ones for doing the work that they need to do. And then the other fraction is progressive motility. So these are the ones that are swimming more or less in a straight line and can actually get to where they want to be, which is really to the egg ultimately in the fallopian tube. So those are the 3 sort of components of motility. But what's also important is the morphology. So morphology is really the shape of the sperm. So the shape of the sperm can determine whether they are actually going to move in the right direction with the right type of motility to get to where they need to be. And there's all sorts of issues with morphology, and in the fact that some of the sperm that you create may have two heads,
That's amazing,
two tails.
Mm.
They may not have a head at all. They may have round heads. And based on the shape of those sperms, we can determine whether they can be functional or non-functional. So there's lots of little pieces of information from all the characteristics of what makes up a sperm sample or a sperm specimen. So to actually work out whether there is the viability of actually creating a baby.
Yeah, I found morphology really strange at first, because I remember going back to my sex ed, you get a picture cartoon of a sperm, and it's got a wiggly tail and a head that's kind of arrow shaped, and that's all we kind of knew. And then I remember chatting to, I think it might have been you or Ian, and talking about kind of morphology and the fact that they can have two heads and they can all be different shapes and sizes. I was like, really? Is this a thing? Is that a thing?
Well, I think what's fascinating with morphology, and this I've had guys that have been sort of knocked sideways with this, is when they read their results. And I mean, this is another part of the whole semen analysis thing, is that nobody actually explains the results to a guy.
What does it mean?
Yeah. Oh, it's normal. Well, what is normal?
Yeah.
Well, and am I just normal, or am I well above normal?
Yeah.
But that'll come in a minute, probably, that bit of the conversation. But morphology is when guys get their morphology and they're told it's 4%.
Yeah,
they're like, 4%, what does that mean? It means 4% of your sperm are the right shape.
Mm-hmm.
Hang on a minute, let me think about that. Let me turn that around the other way. That means 96% of my sperm are the wrong shape.
Yeah.
Mm-hmm.
That's like, for some guys, that's like, oh my God, that's horrendous. No, that's perfectly normal. We are really inefficient, creatures.
Yeah, right.
The vast majority of your sperm, and used to some of you guys, will be the wrong shape.
Yeah.
So actually 4% is normal.
Yeah, and it's actually really, really interesting when you put it on its head like that, because...
Excuse the pun.
As the pun may be. And really what's really important is that of that 4%, you need lots of them. If you're going to get fertilization, you only need one sperm cell to actually fertilize an egg. But in order to run that marathon, there needs to be hundreds and hundreds and thousands and millions of sperm that are running in the same direction, because 4% of a million really slows it down, in terms of the number of cells that are there. So the count, concentration, and morphology is the three biggest markers that fertility clinics look at in terms of giving your overall results. But there are other things that you need to bear in mind.
But of course, a semen analysis it's only a snapshot. That it can shift quite spectacularly between samples, and it does only look at those kind of three parameters, and it's quite a crude index, really. I think it basically, have you got any? How many have you got? Are they swimming, and are they the right shape? That's really all it's telling
swimming
you.
in the right direction.
Yeah, swimming in the right direction. And that's another thing, isn't it? Only a third of the sperm need to be swimming in the right direction for it to be classed as normal.
Yep.
So you've got 2/3 that are either swimming round in circles or not swimming at all.
Yep.
You've only got 1/3 going in the right direction, and you've only got 4% of the right shape.
It's amazing, isn't it? You just assume they're all doing the same job, but actually only a small minority of the sample actually...
I can't remember how many make it to the fallopian tube. It's actually...
it's in the hundreds, I think.
Yeah.
And, it really is a tiny amount. So the normal, natural sort of female genital tract will actually filter out most of the sperm that are not available really for a good, fertilization. So, it's really interesting that you need so many to begin with, and yet so few actually reach the goal.
I think it's also worth just clarifying that kind of the fact that you have a semen analysis, not a sperm test, the difference between your semen volume and your sperm concentration, I think is really important, because your volume, like somebody's volume or something, well, that's actually just the amount of liquid you produce, because it's not just sperm, it's the fluid that comes from the prostate and the seminal gland, isn't it?
Correct.
So,
Yeah.
and then you have your concentration, so it's how many sperm there are in a milliliter of that liquid.
Of that fluid,
Yeah.
Yeah. And everyone goes, is that it?
Yeah.
Yeah.
What 6 million per mil? Or, well, it should be 15 million per mil.
15 million.
Or greater.
It's quite amazing, actually. The numbers are quite quite amazing.
Bonkers, isn't it?
Only one needs to get there. It's like lottery winning odds, isn't it? To get there anyway. It's quite a fascinating science, I find.
But the thing is, with the semen analysis, nothing's really changed, has it? With the semen analysis since it was first, I was trying to look it up just before we started, but I think it was mid-19th century, was it? Some guy in a lab.
It's at least 100 years old. So when we started, sort of counting sperm cells and trying to work out what's going on and how many you need and things like that. But I think what's clear is that the reference values now that we're looking at are based on what they call WHO, or World Health Organization, recognized standards. So those sort of standards now are sort of trying to recognize guys that have a normal, parameters set out, and below those parameters, they're suggesting that those abnormal sort of values should be looked at in a little bit more detail, or that those are the parameters that may need further assistance, like assisted reproductive technologies, ART or IVF type things. So it's really important to understand those numbers. But for me, it's a semen analysis in general terms is quite subjective. There's a number of different methods that clinics often use, and as you said earlier on, Ian, your sperm sample can change month to month. It can change week to week depending on what you do as a person.
Yeah. So therefore, it's always worth doing 2 tests a couple of weeks apart, and certainly where we're offering a home test kit through our store. Yeah. One of them comes with two options, two tests,
minimum 2 tests.
Yeah. So do those 2, 1 week apart, and that will give you your baseline.
Yeah.
But if you've had a fever or a cold or something, a few months before, then that's going to skew your figures quite spectacularly. Yeah. But as we were saying, not a lot's changed in the world of semen analysis.
Not a lot's changed at all. And I think what we've got to remember with sperm is that they are the smallest cell in the body, but they are the most vulnerable cell as well for attack and, being destroyed or disfigured, I suppose, in many ways. There's lots of things that can impair a semen sample, semen analysis. And often if you have got an initial semen analysis that is, impaired, then you should go and get another one and fairly quickly. Yeah.
And of course, what often happens, and we hear this on the infertility support group, isn't it, that guys get the results from a GP, they're told, well, they're normal, or they're not quite as good as they could be, or you've got no sperm, and the guy's left high and dry, isn't he?
Yeah. What next?
Yeah. What next? And what do these words mean? I have no idea.
Yeah.
And they have nowhere to go, and they don't know what to do with those results. And this is why you need to kind of get educated and read up on things, and we're getting distracted by somebody's phone ringing.
Yeah, it's my phone. I've got a new phone on my watch where it tells me when it's beeping away, and I can't switch it off, so apologies.
So there we go. If
Mike was currently getting used to his tech.
Yeah. Oh. Yeah, we're talking about me on another podcast about getting up to speed with Instagram, and Michael's learning how to use his watch.
Like, you guys are all learning. It's great. I love it.
So that's why we're getting distracted and lost our thread a little bit here. Yeah. But the point is, guys go to a GP, they get their semen analysis, they get their results, and they don't really know what to do after that. And if they're suboptimal, again, they don't know what to do. But also, that really is just a basic test, and there are other options open to men. I think before we go down that road, I think it's just worth pointing out we've got a whole news article on the website all about understanding your semen analysis, so it's worth having a look at that if you haven't checked it out already at testhim.com. But there are other tests that really... Oh, it's the phone again.
Yeah, no, it's voicemail. Okay.
There are other tests available that actually give a much better indication of a man's fertility potential, really, don't they?
Yeah, there are.
Toby, you alluded to this as well, that's what made the difference for you on your journey, was some of these advanced tests.
Yeah, exactly. And if we just rewind just a touch to the semen analysis, so my count was always in the right range. My motility was okay, I was told, but my morphology was always at 2 to 3%. But I was just told it was okay, and I wanted to understand a little bit later down the line why. And that was the thing, I guess, really that I questioned. Could I find out more about it? And that, I guess, now leads us on to the next set of assessments and what's available.
Yeah, which aren't routinely available through the NHS, unfortunately. And even some IVF clinics aren't running these tests. But Michael, you're probably the leader in terms of getting these tests out there, through your other company, Logix. What are these other tests? What other things should guys be aware of and consider?
Well, I think just to put it into perspective, the semen analysis, the standard semen analysis, is conventional, and it's the route to get most of the analysis done in the first instance. So we can't, I'm not going to rubbish, the standard semen analysis, but we know we've got different methods to assess sperm in slightly different ways, sort of enhanced or advanced ways, if that makes sense. So as we said earlier on, sperm cells are very, very vulnerable, and one of the things that they're very vulnerable towards is attack from a reactive oxygen species. And what that is simply is oxygen radicals that are floating around in our body, and if they're in excess, they can create chemical reactions which actually impairs the sperm. And so reactive oxygen species is a very well-known phenomena, which contributes in many ways to what we call sperm DNA fragmentation. So this is where the DNA integrity of the sperm becomes impaired. And there are a number of different assays or different tests that can be run to look at these reactive oxygen species or the impact of sperm damage that don't necessarily normally get done certainly at NHS level or GP level, and certainly doesn't necessarily get done at secondary care level in IVF or IVF clinics. So those types of tests are things like directly measuring oxidative stress, and tests like the oxidative reductive potential test, or MyOxys test, often referred to as an SOS test or sperm oxidative stress test, is one way to assess that. And it's quite a simple way of assessing the sperm. You basically have a sperm specimen, you put it on a little electrode, and you're looking at the resistance, really, of a current flowing through the sperm specimen. And it gives a millivolt reading. So it's like a voltmeter, really, for your sperm in many ways. But what we know is that there are levels at which where the reactivity of the sperm or oxidative stress within the sperm, if it's above, say, 1.34 millivolts, I'm going to say a lot more words as well, millivolts per million sperm per mil.
That's a mouthful.
So it's a mouthful. It's a massive mouthful. But effectively what we're doing is measuring the voltage, dividing it by the concentration of the sperm, and it gives us a reading. Now, if that reading is above 1.34, it's determined that you are in oxidative stress, or your sperm is in oxidative stress, which does reduce the viability of being able to conceive either naturally or through an assisted reproductive program such as IVF or ICSI. So what we need to think about is what is the cause of that and what do we do with it. So that's one method of measuring it. There are other direct measurements of reactive oxygen species and assays that are called chemical luminescence, another big word, but basically we're looking at the light emitted through a chemical substrate that we put the sperm through, and based on the light values, we know whether there is this reactivity that's going on.
Is that the Ross test?
That's the Ross test,
because that's what you had, wasn't it, Toby, at one point?
Yes, I did.
Okay.
And I think that actually come back relatively normal, I was told at the time, but I think that got worse over time.
Yeah.
But I'll come back to that in a second.
Yeah.
So we've got MyOxys, which is an oxidative stress test.
Yeah, and that's really, really simple. There's a Ross test, which is normally done in an external lab. So
and both of those are really kind of measuring the same thing, but just in a different way.
Similar aspects of the sperm reactivity, in terms of the amount of oxidants that are present in a seminal fluid. And then the other type of method, and there are 2 or 3 main assay types, but DNA fragmentation is the other type of test that you can do, which is really looking at how the base pairs of the sperm DNA integrity are structured. So what we know is that you can get single strand breaks, double strand breaks of the double helix. And
just to clarify, this is different to the oxidative stress test now, isn't it? This is a different type of test.
This is a different type of test.
Just looking at the DNA integrity within the sperm.
So what we're looking at is really the end point of the damage done by the reactive oxygen species, because what we've got is a sperm cell which has been under attack, and then you have got this sperm cell which has got DNA in it, but it may be impaired. It may be impaired for both fertilization, blastulation, and actually creating an embryo. So we need to look at what is the quality index of that. How much has that structure been damaged? And that's what we call DNA damage, or DNA, sperm DNA fragmentation. So fragmentation does have a direct relationship with the outcome of trying to to have live birth rate and have children, basically.
Yeah. So far we've got DNA. So we've got semen analysis,
Yep.
oxidative stress test, DNA fragmentation testing.
Yeah.
What else might a guy want to consider? Are there any other tests that they could look
so
at?
it's really interesting. So what we've got to think about is not just the sperm itself, but the factory of creating sperm. And the factory of creating the sperm is under the influence of our brains, funnily enough. So blood hormones are responsible for triggering the response to the testes to say, come on, guys, get working and produce some sperm. But they also produce testosterone. And there are sort of 3 or 4 main hormones, blood hormones, that are really important to check. So those types of blood hormones are things like follicle stimulating hormone, luteinizing hormone. These are often referred as female type hormones.
Both of those.
But those are produced by the pituitary gland and in the hypothalamus, and they trigger a natural sort of negative feedback mechanism to the testes to produce testosterone. So they have direct trigger onto the Sertoli cells in the testes, and then they will then create testosterone. Testosterone is a negative feedback loop, so if there is enough being produced,
that's a break on the production.
it slows down the amount of follicle stimulating hormone, and the loop sort of continues. So there's about 3 or 4 main components that should be looked at, and if they're out of whack, then clearly there is either going to be a reduction in the amount of sperm being created, or there can be abnormalities in the shape of those sperm being created because they're not getting the right signaling, they're not getting the right hormone elements to it. So, and that can also be an indicator of other bodily state diseases.
So we're actually quite complex creatures, really, and we've got all these hormones flying around. Funnily enough, I'm still trying to write the news article on hormones and male fertility, so it will be on the website at some point. Hopefully by the time this comes out, it'll be on there. But I'm trying to get it into the simplest terms and help people understand, because it's like, we don't think about it. You don't get up in the morning and think, right, what's my FSH and LH doing today? Are my testosterone levels okay?
It's not something you think about at all.
What are my testicles up to today? Are they producing enough? LH or whatever it is, or testosterone. But it just shows that there's so much more to male fertility than just a semen analysis.
Well, it's really interesting when you look at hormones. It has an impact on our general health. So things like stress. There's a cortisol. That's a hormone that gets released. That can have an impact on sperm cells. But also things like libido and awakeness, alertness, and strength and exercise. We can all be impaired by having a dysfunctional hormonal profile. So it's really worth getting that done, getting it checked, and if it's abnormal, then you need to see the right physician that can correct it for you.
And I guess that's probably going to have to be towards the end point here of this conversation is, okay, you can get your semen analysis through GP, but all these other tests, unfortunately, aren't routinely available through the NHS, and that's I think that's an evidence thing, isn't it, and a kind of funding thing and what have you. And part of what we're doing with testing is to try and change that and get this testing available for men as routine. But I guess for a man, it's like, well, okay, it's great, I've listened to this and now I've learnt a bit more. What do I do now?
Yep.
And where do I go?
Well, I think you've got to ask the questions, really. So if you're on your journey, whether you're at the very start of it, if you haven't had a semen analysis done, go and get one done. If it's abnormal or it's you're still struggling to conceive and it is normal, then you need to go and get a referral. And that referral will be typically to an IVF centre, and then it's a matter of just working out what could be any probable cause of whether you've got any abnormality, whether you've got low count, low motility, your morphology is not quite right, and just try and think of isolating what those sort of causes and consequences could be. But what we're trying to do is actually ask those questions. Is that what is that probable cause before you've actually had a semen analysis? Because there's lots of questions that often don't get asked in GP land that actually could help you assess your risk. And that's what we're trying to do here, isn't it?
Yeah, exactly. And I suppose that comes full circle back to me in the beginning part of this story, really, because those questions were kind of asked to me. One of the first things that was asked was, have I got any lumps or pain in my testicles? I remember having that conversation with you, Ian. And it turns out that I had a varicocele. So therefore, I went off to have a DNA fragmentation test done to actually just see if that varicocele, which, let's explain, it increases your temperature of your testes, which ultimately makes the factory too hot, doesn't it? So they don't get manufactured as good. And it came back that I had very, very high levels of DNA fragmentation test, which was obviously very difficult news at the time to understand. But that knowledge was the fact that it was caused by my varicocele, which meant I had to go and have some surgery. Scary surgery, but it wasn't. It was dead simple. Dead simple. And I then had to wait 3 months for the recovery of that surgery, to be retested and see if the DNA improved. But I landed on the good side of the coin and
didn't need that test.
Didn't need that test.
Didn't need the second test.
Yeah, which is
proof was in the pudding.
Still blows my mind. But in the topic of male fertility testing and why this is all important and why we're giving you this information in this podcast is that a basic semen analysis gave me an overview of my sperm health, if you like. It was okay to see, okay, I've got numbers, I've got them swimming in the right direction, but perhaps some of them aren't shaping correctly. But the DNA fragmentation test gave me this new information that actually there was something underlying going on, that there was an issue, and pretty much no matter how much IVF or ICSI I'd have had, probably would never have fallen pregnant because the problem was so bad. But then a urologist was like, this is what you need, a varicocele repair. I went off to have that done. The rest is kind of history, really. But
I think the point there is your semen analysis was pretty much normal. Wasn't it?
And that's it. And that's it, isn't it? And that's the scary thing, really, is that the sperm looked okay, but actually the DNA inside of them are pretty rubbish. It's a bit like having a really nice Ferrari with a broken seat at ECU. It'll be parts on someone's driving.
Ford engine in it.
That's really nice.
You just can't see what's going on under the hood, can you?
Exactly. And it takes a specialist to look at that and see what's going on. And I think I always go to the analogies of motorsport.
Of course.
But that's the way I looked at it. I was like, okay, well, I've got them. But if the DNA is no good, how can I improve them? And there was thankfully a really simple...
Not firing on all cylinders.
Exactly that. But on the topic of you just mentioned about kind of asking the right questions, as part of the Test Him campaign and what we're trying to do, if you head over to our website, we've got a portal that you can register for free. You can log in, and we do have a free assessment questionnaire that can just start highlighting to you of the risk that you might be at, that you might be suffering from these conditions, such that I have. And it will point you in the right direction of what to do next, and advise, which tests to go for, because I think it's really important at any phase in your fertility journey to constantly increase awareness of your own health, empower yourself with knowledge that we're hopefully be able to provide to you, but then get you the correct treatment pathway that you really need to be on, or actually that you're okay and crack on as you are, and find that maybe IVF is still required or whatever. But yeah, head over to our website. If you listen to this on your phone now, hit pause and head over and have a little butchers, because I think you'll find it be really, really beneficial. It certainly was for me. I think the portal's kind of been designed from the three of us, our journey to this point. We've all chipped in of kind of what is needed in this space, and go ahead, have a look, and
get tested.
Get tested.
Yeah.
Get checked. Cool.
Testhim.com. Dead easy.
Dead easy. Yeah.
Cool. There we go.
But I suppose that wraps up this episode.
It does.
Another episode of the podcast. This feels really nice, to be fair, doesn't it?
It does.
Yeah, and I hope you guys enjoy tuning into our podcast. And come along next week, and we'll have some more information about
next week,
next month, next week,
whenever it is,
when the next one is.
Yeah, when Michael's turned his watch off.
Yeah, maybe.
I've got to work that out. Sorry about that, folks.
Yeah. Cool.
All right.
Brilliant. Thanks, everyone.
All right. Take care, everybody.